science friends. We are going into our last week of gene regulation, so we're going to finish out the last few lessons here this week, and then we are going to be gearing up for a test early next week. So get kind of in the mindset for that. Start reviewing if you haven't. This is a pretty jam-packed unit. And then we're going to talk today about several different things that are regulatory mechanisms in RNA. Now, not all of this is really testable content, but I just want you to see that there are so many things that happen that help to regulate our genes, what turns our genes on and off, kind of continuing this conversation of how gene expression is regulated. So I will go ahead and point out the parts that are really important, and you may want to star those in your notes so that you especially remember to look over those parts before the test. So this first part that we're going to talk about today, alternative RNA splicing, is definitely one of the important parts. So put a star here to review this part about alternative RNA splicing. So normally in a gene, remember, so this is an example of a troponin gene, and it has several different exons in it. And so alternative RNA splicing basically is picking and choosing which of those exons you want to use to be able to make a protein. Okay, so in this one gene, we can transcribe, this is just showing two options, but there would be many more options of mRNA from the one gene. So this one has exon 1, 2, 3, and 5, and this one has 1, 2, 4, and 5. And so those are going to produce different polypeptides, different proteins based on that mRNA, based on which exons are spliced in and which ones are cut out. And so when we look at the human genome has, compared to a lot of other genes, and especially for its complexity, has a relatively small number of genes. So the genes in the human genome is actually comparable to like a mustard plant or like an earthworm. So relatively small number of genes. A fruit fly has a lot more genes than in the human genome. But because of this alternative RNA splicing, we have a lot more protein combinations that contribute to the human complexity. So this is really important as far as making different proteins and having lots of more options in our gene. So let's talk about a few ways that genes are regulated. So this is post-transcriptional. So after we've taken that DNA and made it into mRNA, then we're going to regulate it somehow. So the first way is through mRNA degradation. So the mRNA can, if it stays active and it remains in the cytoplasm, it remains at the ribosome for a long time, then it will be translated more. If it's degraded pretty quickly, which generally it is, specifically in like bacteria, they're usually degraded within a couple of minutes, then it's not going to be translated into many more proteins. And so that is one way that we can essentially either keep producing more proteins by allowing it to remain, or discontinue the production of proteins by causing the mRNA to be degraded. And ultimately, it's determined by a nucleotide sequence in the UTR, so in the untranslated region at the 3' end. Remember, we talked about that 5' cap and poly A tail being really important in them being able to bind, and that's what we're kind of getting into here. By them being able to attach on the ribosome and be able to bind. So if we initiate that translation and those are attached, then translation will happen. If those are not fully constructed, if that 5' cap and that poly A tail are not there at the end of transcription, then translation won't be able to occur. And so this happens in the egg cell. So normally the egg cell, before it's fertilized, we don't need those genes to be expressed, right? And so it's that poly A tail isn't sufficient in length. But after it gets fertilized by the sperm, then that poly A tail will continue to be added onto. And those translation initiation factors will be activated, and so then these the mRNA, the regulatory proteins, those translation, that's the same thing here as the translation initiation factors, will be activated, and so then translation can proceed. Okay, so if we stop the mRNA by degrading it or block the initiation of translation, that's two ways that we can regulate gene expression. A couple of other things: chemical modifications to polypeptides to make them functional. So that's an example. So, some of these polypeptides have to have a sugar or a whatever attached to them at the rough ER. And so if that doesn't happen, then that's going to make that polypeptide not function. Target destination. If it doesn't reach its target destination, like if it's supposed to go to the liver cell, but it hasn't got to the liver yet, then it's not going to function properly. If it's supposed to be in the membrane, but we don't get it to the membrane, that's another way that it can be regulated. So regulation can happen at any of these steps. Length of time for the protein to function in a cell. So sometimes proteins are active temporarily, right? You don't need them continuously and for a long term. And so the way that this part's really important too, in addition to RNA splicing, put you a big star right there. One of the ways that proteins are marked for, or the way that proteins are marked for destruction, is ubiquitin. You may put a square around that as well. And so that is attached to the surface of a protein and causes it to be degraded. Okay, it's kind of like apoptosis. Remember, that happens at the cellular level. Ubiquitin and degradation by a proteasome is what happens at the protein level. Okay, so ubiquitin is attached, and then there's a enzyme, so to speak, called a proteasome that will bind to that protein and cut it up into these little fragments. Okay, and so that will help to regulate protein function as well if we get rid of the protein, obviously. Okay, so when we look at our DNA in humans, of our DNA, only about 1.5% of it codes for proteins, and so that's a very small amount. A little bit more of it, we have a lot of non-coding DNA. Some of that's going to go to make the rRNA, the ribosomal, and the tRNA, the transfer RNA. But then a lot of it is going to be non-coding RNA, which is abbreviated here ncRNA. So let's talk just a little bit about this non-coding RNA. So one example of non-coding RNA is microRNA, abbreviated miRNA. And so microRNA, what it does is it binds to complementary mRNA and causes it to prevents translation or causes it to be degraded. So when it binds, so here's our mRNA here, miRNA, our microRNA, and it binds to the regular mRNA and forms this structure called a hairpin structure. And then there's this enzyme called a dicer that essentially kind of cuts it up. And so either the mRNA will be degraded, or when it's bound like this, then translation is going to be blocked. Okay, you're not going to have translation of that protein. So again, this is coded for by the DNA, this non-coding RNA, but it's important because it's involved in regulation. Okay, so you all watched that epigenetics video on Friday. Hopefully you found that really cool and interesting. I think it's really cool and interesting. And so this one, this is one of those things how, identical twins could be different because of differences in non-coding RNA and differences in ways that things are turned off and turned on, and so on and so forth. So this process of injecting that double-stranded RNA molecule, like we saw here, forming that hairpin structure, is referred to as RNA interference. And so that's going to turn off the expression of the gene with the same sequence as the RNA because it's complementary here. And so that's going to turn off that gene expression, and that's called RNA interference. Okay, so that's pretty short and quick, but that wraps up today's lesson, and you all have a great day.